First thing on the agenda, given a few different factors, we are going to have a seating chart for you on Monday. Okay, maybe not everyone will change seats, but there will be a shuffling. So just make sure you're aware of that. If you guys have a serious special request, such as I need to be in the front, I can't see, then let us know. But it's not going to be like, oh, let me sit next to my friend, because maybe you will, maybe you won't. Okay, we've been observing and we think there's a couple changes that need to be made. So expect that on Monday. We're going to continue with notes and the 3 types of stem cells. We kind of went over it, but let's all say it together. The one that starts with a T is what? Totipotent. The next one down, pluripotent, and then multipotent. All right, what is the difference between these? Well, this question kind of gives you a preview to that. Which one can differentiate into the most type of cells? Totipotent. Toti should remind you of maybe what kind of word? Total, like all of them. Okay, so totipotent stem cells can turn into literally anything. Okay, potent, what word are we thinking?
Smelly.
Smelly, like potency, right, is the strength. So the strength of a totipotent is like totally strength. The next one down. Toti is the most, then pluri. Okay, pluri. What kind of word are we thinking of when we say pluri? Plural. Okay, so plural is still like multiples, right? So pluripotent is a little less strong than a totipotent. And then the last one, multi. We can think of multiple. So that one's kind of hard because we have plurals and multiples. That's like a synonym, right? But at least the total reminds you which one's on top. Is this in backwards alphabetical order? Okay. Sometimes a study skill is noticing that a pattern is like in alphabetical order. This one's in backwards alphabetical. So you can remember that T, you go backwards, M, it comes before P, comes before T. So that's just a trick. You'll actually be surprised how well that works. You'll be like, I have these 3 potents, which is the order. And you can remember, oh, it's backwards alphabetical. And then you know which order it goes. Okay. All right, so if you don't have your notes done, you won't have time to do it now, but I'm just going to kind of quickly go over and make sure it's clear what you should have done in your notes. Okay. With also toti, pluri, and multipotent, you want to know where these things come from, okay? So a zygote is totipotent. A zygote is like the very beginning of you, okay? Right when sperm and egg come together, pretty much right after that, you get a zygote. The next one, pluripotent, comes from something called the inner cell mass of a blastocyst. So in the next picture, you'll see a blastocyst. A zygote, after it divides multiple times, it becomes a blastocyst. Okay. Then, way further down the line, multipotent can come from umbilical cord stem cells. That's, you get an umbilical cord. I don't know at what week that happens, but it starts developing right away. So you can get stem cells from there. This just kind of goes over the same thing, but some interesting facts. Totipotent can give rise to literally any, and in case you didn't know, you have 220 different cell types. Pluripotent can basically give rise to all the cell types except placenta. So pluripotent are still really good. Okay, they're very efficient to use for regenerative medicine purposes. Multipotent is limited. Okay, so we'll look at those examples. This is just kind of a helpful study tool. You don't need to know the genes. Okay, that's not something you would need to know. But these examples, this first part and this bottom part is going to be helpful. So this is a really great table for you to study. Pros and cons of use of research. We're going to actually go into that. So great study tool right there. Okay, this is just kind of showing you how the pluripotent embryonic stem cells eventually differentiate themselves, right? This whole unit is about differentiation, your cells becoming specialized. So pluripotent will differentiate at some point into these 3 types. You don't have to memorize those 3 types, which will eventually get you into multipotent stem cells. So notice that, for example, this yellow group can now only turn into the lung and pancreas cells. Okay, so if we use these yellow stem cells for study purposes, we can only use them to study lung and pancreas issues, right? So they're more limited in what they can become. So that's what multipotent means. All right, you should have annotated this figure. You're looking at your notes and there's nothing written on this picture. You need to go back and do that, okay? So notice that each box kind of has a different color just to make them look a little different. But this is exactly what we were seeing in the first part of the notes. We have our zygote, which is sperm plus egg, you get zygote right away, very beginning. That turns into an embryo. And notice this embryo here has an inner cell mass. Okay, that's basically just a bunch of cells that are quickly dividing, which will eventually specialize into a human body. But these, the inner cell mass of an embryo is pluripotent, okay? So this is showing that if we take a cell from this inner cell mass, that is a pluripotent stem cell, which can then actually turn into any of these multipotents, okay? And then purple on the end. This is kind of showing in terms of the research side, we can actually harvest these stem cells. We can get stem cells from the embryo and we can grow them for use in however we want to use them, okay? Remember, a zygote might be a little more difficult, right? To catch a zygote, the technology and the ethics that goes along with that is very tricky. A little less tricky with the embryo. You can see an embryo, it's a more stable structure, it has a lot more cells, and you can actually take out a pluripotent stem cell. And then down here in the bottom, we can actually take adult stem cells, and those ones are going to be the easiest ones to get. Like I can get a stem cell right now from your bone marrow, and you would be not too badly hurt, right? It might hurt you a little bit, but you would be healthy. So I can take stem cells from your body right now. No big issues. Well, most of these people are donating. It would be illegal for me to take it otherwise. Okay.
This means that even though they contain the same DNA, a skin cell will be different to a liver cell. To work out where they all come from, we need to go back to the start.
We all start life the same way, with a sperm cell fusing with an egg cell to create an embryo. As the embryo develops, it divides, creating a whole bunch of undifferentiated cells which then go on to form every part of the body. These are the embryonic stem cells. These stem cells are the master templates, which have the potential to become any type of cell in the body. So, understandably, scientists around the world are pretty interested in them. Embryonic stem cells can be collected from donated early-stage human embryos, once they form a ball of around 70 to 100 undifferentiated cells, called a blastocyst. They can then be taken, grown and maintained in cultures in the lab. In May of 2013, scientists in the US managed to generate them from adult cells, using a process called somatic cell nuclear transfer. Somatic cells are the cells which make up the body, such as liver, brain, blood, or skin cells. It's the same technique that was used to make Dolly the sheep. But how did they do it? The team started by taking an unfertilized human egg and removing the nucleus, which contains the DNA. They then took a skin cell called a fibroblast and inserted its nucleus into the emptied egg cell. Within 30 minutes, the cell had fused to generate an embryo. It's then zapped with electricity to kick off the growth. The fluid inside the cell, called cytoplasm, reprograms the DNA from the skin cell and allows it to return to a stem cell-like state. This means that the cell divides just like a normal embryo, producing stem cell after 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 stem cell. So, what can they be used for? The potential medical applications for this type of technology are widespread. Using neural stem cells, which generate brain cells, show that it might one day be possible to repair some of the damage caused by a stroke. Scottish researchers are currently working to develop synthetic blood cells, which could provide a new source of human blood for patients and reduce the demand on blood donors. And in future, scientists might even be able to grow replacement organs in the lab. If cells can be taken from a patient, then they can generate stem cells and grow a tailor-made replacement. Pretty big things from such a tiny little cell.
Okay. Notice that that video was from what year? 2013. So all of these things, he's like, oh, these scientists are working on this, these scientists are working on that. Might be interesting for us to go in and see, have they done it yet? What's the progress bit? And I wanted to take a moment to mention, if you haven't seen today's Remind, it is an article. The article is titled Cartilage 3D Printed from Stem Cells to Replace Body Parts. So basically in this 3D printer, they put stem cells into the printer instead of the regular plastic or whatever they use. And then the printer was able to print out those stem cells and make a shape that is human cartilage. So I guess you could call it like a bioprinter because you're literally putting cells as if they're Legos into a printer and it puts them together. And now you have a tissue. So you could, I know, it's crazy. I posed a question in the link and I will give some extra credit to the first person to give me a good answer. That would probably include a resource of some sort to my question. So go ahead and read that. Not right now though. But I just wanted to say that they were able to get pluripotent stem cells. It says they used cartilage cells taken from humans in connection with knee surgery. So when they're in the knee surgery anyways, they just scoop out a little bit of cartilage. And they reversed their development. So they took out the cartilage cells, they reversed the development to turn it into a pluripotent cell. So they took, it seems to me the way it's written, they took a multipotent cell and took it backwards in time to where it was a pluripotent cell. How they did that, I have no idea. So definitely read the article, it's super interesting. And towards the bottom, it poses a problem. The article was written in 2017. So I would like you guys to maybe see if they have solved that problem yet. Okay. All right. So I read that article, but I haven't done research to see if they fixed it. Mainly I was on the way to work and I was like, oh, I wonder if they fixed it. And I was like, I don't have time to look it up, but maybe my students do. I put that in the text because I have a million things to do right now. Okay. This kind of just shows you the same thing. Why they are good for research. Okay, they divide repeatedly, so you can have unlimited of them, the stem cells, and they're not differentiated. You can turn it into whatever you want. It's like a piece of clay, right? You can mold it to whatever you see fit. Okay, so our first piece of notes that we are filling out together today is the use of cells to treat Stargardt's disease and one other named condition. So we're going to go over Stargardt's and leukemia and the use of stem cells to treat these diseases. Okay, so it says the capacity of stem cells to divide and differentiate makes them very suitable for therapeutic uses. Researchers and doctors hope stem cells can help us to do three things. And the red is your first one. Increase our understanding of how the diseases occur. Should be writing this under that first bullet point. This would lead us to kind of a preventative measure, right? Preventative measures usually are the best. Stop the problem before it even happens. So if we can figure out how these diseases are happening, we can figure out maybe how to stop them. Okay, so increase understanding of how diseases occur. Next thing, generate healthy cells to replace diseased cells. So if we can't do the preventative measure, we could at least try to use these as a regenerative piece. We can give you healthy cells to replace your unhealthy cells. So on the image there, it's just showing some of the areas of regenerative medicine applications. Our first one, Stargardt's, is going to have to do with the eyes. And our second one, leukemia, would have to do with the blood. But you've got also your brain, heart, and muscle areas on there as well. Are we feeling a little better about the $5.5 billion? Was it billion or million? $1 billion that we just put into more stem cell research? I want you guys to think about that. If you could have voted on Prop 14, what would you have voted? So keep thinking about that as we go through the unit, because your opinions may change. I did vote. I'm actually not supposed to tell you guys how I vote. That'd be a lot of car washes. Okay, you probably can guess how I vote. Test new drugs for safety and effectiveness. So instead of testing drugs straight on people, like we saw in the last unit or 2 units ago with the antibiotics, they're like, oh, a dying woman, let's just use her as our guinea pig. Instead of doing that, we can test it just on cells, ethically, way more safe, right? Make a lot less people mad. Liver damage? That might be a source. I'm not sure if it is true. I would assume if you are a donor, I would assume that you are also willing to donate your stem cells. And also, as a human, as an adult human, you don't have very many pluripotent or totipotent cells. There's very few of those left, so they're very hard to get. You would have mostly multipotent cells, which can be useful, but not as useful, right? That's why they're focusing their research on embryos, because they get more useful things from embryos. But it doesn't mean they're not taking stem cells from donors as well. That's a good question. Okay, here is a video where you're going to see what Stargardt's disease is. I have a slide on this, but you could take from the video cause and symptoms as well. Okay, so you need to know for your syllabus statements, what is this disease and how do we use stem cells to treat it? You can turn off the lights, somebody.
11-year-old Megan's parents had never heard of Stargardt and initially thought their daughter only needed glasses. Then they received her diagnosis.
For us at that point in time, it was devastating. It was so difficult to hear that your child was, diagnosed with a condition that was going to affect her vision, and she would be legally blind, and at that point they were words. We really didn't understand what it would mean for us or for Megan.
The toughest thing about having Stargardt is that you can't see exactly everything that everybody else can see.
When Megan was diagnosed, her eyesight was 20/50. It has rapidly deteriorated to 2200. But large print books and vision-enhancing devices allow Megan to learn at a normal pace and remain in the same school she has always attended. Dylan is a high school student who also has to deal with the challenge of Stargardt. His school, however, is providing priceless support.
They have helped Dylan from the onset. They called in specialists. I guess it took about 2 months to get everything in place, get his equipment ordered. He uses a CCTV, and all of this was done by the school.
When I'm sitting in class, if I don't use my machine, my CCTV to see the board, I can't. It all just looks like a blur and I can't see nothing. I can zoom in how close I want to, zoom out to find things around the classroom.
The school also provides an aide for Dylan, and the state provides a specialist who comes in to work with Dylan once a week to make sure he's using his assistive technology and vision-enhancing equipment properly.
That device allows him the same opportunity that every child in the room has. Nothing really changes with a visually impaired student in the room. Having a student like Dylan in the room is just the real world. It's life.
Crystal is 49 years old. She was diagnosed with Stargardt in the 3rd grade when the teacher told her parents that she had cried in class because she had trouble seeing the blackboard. Her mother took her to an ophthalmologist.
When they flip those little things, does it look better this way or does it look better that way? All she could see was the big E. Then the ophthalmologist said to me, she will not go totally blind, which was kind of a shock because I just thought she needed glasses.
Soon thereafter, another shock. There is a genetic component to the disease, and Crystal's brother was also found to have Stargardt. Today, Crystal works at the Boys Town National Research Hospital in Omaha as a genetic counselor and vision program coordinator.
The fact that Crystal was legally blind was not a consideration at all when we hired her. She had so many capabilities and skills. Crystal has been an outstanding employee from day one.
Monday, November 1st.
Crystal uses assistive technology to help with her work. A computer with a speech synthesizer helps her read information on a screen. And she uses a small camera and closed-circuit TV to enlarge material up to 60 times.
In my laboratory at UCLA, we're working on a mouse model of recessive Stargardt disease, and we have seen very promising effects with a drug, and this drug will be entering clinical trials in Stargardt disease patients over the next year or two, and we are optimistic that we will see a therapeutic effect in those patients.
People often talk about your attitude being more important than the ability. And I think that Crystal had a good attitude about her blindness and was able to accomplish a lot of things because of that.
The biggest, I think, and most important thing is just to be as comfortable with yourself as possible. That gave me the self-confidence as an adult to be able to raise my children and have a normal, happy life.
So, you have a little bit of an idea of what this disease is. Let's go ahead and turn those lights back on. Yes. Okay, here's your cause. So they touched on the video saying that there was a genetic component to this disease. So you may not know a lot about what this means yet because we're actually getting this in unit 16, I believe. We'll learn more about inheritance of disease patterns. But for now, we'll write down that it is a recessively inherited disease. You guys remember that there's dominant and recessive alleles? So this is caused by a recessive allele. And it is caused by a mutation in a specific gene called ABCA4. So when we look into DNA and mutations and diseases, we'll connect back to this. Also, it does say it's autosomal up here. That means it's not sex-linked. You guys remember from freshman year, we looked at sex-linked diseases like color blindness. This one is not sex-linked. It's in one of the other chromosomes. Okay. We're only writing the red. Okay. We are specifically looking at this, obviously, because it's a stem cell treatment example. We can see that the worldwide prevalence is 1 out of every 8 to 10,000 people. It's pretty low, right? How many people do we have in the world? 7 billion. Actually, I haven't counted recently. Do we know? What's the most recent number? I remember when we hit 7 and the world was like, ah, we hit 7 billion and we're still climbing. Yeah. Okay, Stargardt causes progressive vision loss. This is your symptoms page. Or box. Progressive vision loss beginning in childhood or adolescence, and it's mostly the central vision. Peripheral vision remains intact. But if you had to choose one to go away, the peripheral vision is probably a little less important than your frontal vision, right? Imagine walking around the world and this is what you see. Black, and then you see a little bit on the side. I feel like that would give me a headache because I would constantly be trying to like see the outsides, right? That would be frustrating. Yes. Oh, good. Yay. We'll have you guys announce that in our meeting today. Interact Club today at lunch, by the way. Interact Club.
Yeah, glaucoma.
Huh? Yeah. Yeah. 7.6 billion. Wow. Man, we're on our way to 8, guys. This was 2019. Wow. I wonder what we're at right this second. Yes. Oh no, we have a new number. 895. When was that? Today? Oh man. +1. Plus Zachary. Zachary will be his name. He's coming in 4 weeks or maybe less. I have liked it up until recently. Now it's getting hard. All the time. Yeah, all the time. I still enjoy it, but things are definitely noticeably more difficult. Okay, so this is what they do. You don't have to write this. We do want to explain it actually. Okay, shh. So you have this picture, but I think we should add a couple notes to this picture because it says to explain. On a test, drawing a picture is not good enough for explain, right? Okay, so stem cells can be coaxed to grow. You don't have to write this whole paragraph. Let's just take out some key points. All right, stem cells can be coaxed to grow in any type of cell or tissue found in the body. In the case of treatment for Stargardt's disease, human embryonic stem cells are differentiated to become retinal pigment epithelium, RPE. I would say that's important to write. A cell layer lining the back of the eye which nourishes and supports other cells. So we know what they're growing and what that cell does. To grow, I would say it probably doesn't take very long, but I don't know. I'm sure they know. I don't. In human trials, the implanted RPE cells have restored the central field of vision. So then implanted RPE cells have restored. You could just put central vision if you want. That's a good question. That would be awesome for somebody to look up. Currently, the treatment of macular degeneration with stem cells is in the early phases of human trials. This would be another thing to look in on. I came across this information 3 years ago and I haven't checked up on it yet. That could be maybe another piece of extra credit. Somebody wants to bring me like a current piece of news on treatment of Stargardts. That would be awesome for you to share with us. Yes. Yes. Uh-huh. Oh, really? Yay! They're trying to bring back the northern white rhino because it went extinct. But before they went extinct, they were able to save some embryos. Yeah. I think in our freshman year, we were together when the last one died. Really? Yeah, I think one of the ones was the pink dolphin that's in the Amazon. I think they thought that was extinct and it came back. So that's good news, but I'm sure it's still like severely endangered. Okay, are we good? Yeah, it's like the pink river dolphin or something. And it's a freshwater dolphin because the Amazon. Okay, leukemia. So let's look at this. Outline the cause of leukemia. Leukemia is an example of a disease that has been treated using stem cells as well. Yes. Oh, this is page 9. Oh, you're so, thank you. So this is where it's going to get annoying. You can pass them out. Some of you may want to unstaple and restaple because this should go right after the page we just went over. You're going to get that off. But chart will change, so maybe someone else. I have a staple remover if you need it. I have 2 staple removers. How lucky. Pass it around. It should go right after this current page. Do you guys want to go over page numbering? Because that was my fault, not you guys. Yeah. I did number unit 6 now. All right, let's pause our recording because stapler. Oh. Okay. While we wait for that, let's keep going. So leukemia is an example of a disease. We could do it quietly though. That has been treated using stem cells. It is cancer of the body's blood-forming tissues, including bone marrow and lymphatic system. People with leukemia, so wait, the cause. Okay. People with leukemia produce excessive and abnormal white blood cells called leukocytes. Those white blood cells are there usually to fight infection, but those leukocytes can begin to grow and function abnormally, becoming cancerous. Okay, I'll come back to this. Give me just a second. Just want to make sure there wasn't anything on these slides. Okay. So in terms of like what causes leukemia, I don't think they have that answer yet in terms of like is it genetic or is it environmental? Some diseases we just don't know at this point. Okay, my friend had leukemia. Nobody in his family ever had leukemia. They don't know what started it. Yes. Yes, it is normal for a lot of cancer patients because it is actually the cancer treatment. Alan, please close that down. So, yeah, it's part of the treatment actually. It's not the cancer that causes hair loss, it's the treatment of the cancer, the chemo. Yeah. All right, so the cause, you're writing the bold here. Sorry, it's not red like usual, but it's the bold. It it is excessive abnormal white blood cells. And then those blood cells become abnormal and cancer. So successful treatment for leukemia depends on getting rid of the abnormal leukocytes. So they usually do this by chemo. They use potent drugs to target and kill the abnormal cells. After chemo, a bone marrow transplant can be used to replace the multipotent stem cells. So when it says explain how stem cells are used in the treatment, you have kind of a little image there. There you go. You have some things that you can write down here. So first, they have to kill the bad blood cells. I would put that somewhere. First, they have to get rid of the cancerous white blood cells through chemo. Then we have to replace them. So the stem cells are specifically coming in handy when replacing what we got rid of, right? If your blood is like 75% bad cells and I take out all those cells, now you are severely lacking blood, right? So we need to replace them. So bone marrow transplant gives healthy stem cells introduced into the patient's bloodstream. This would be the process of finding a match, right? It's just like an organ. You're getting a heart transplant, you have to make sure the blood types match and other things. It's not just blood type. So if they find a match, then they can take bone marrow from that healthy human and insert it into the sick human. If the transplant is successful, the stem cells will migrate into the patient's bone marrow and begin producing new healthy leukocytes to replace the abnormal cells. Kind of like a factory. If one piece of the factory is not functioning, you're going to take out that piece and put a new part in whatever machine, right? All right. When is our test? Our test is Wednesday. Okay. I'm just trying to decide what I need to do. I'm going to give you no homework today, but you may have homework on Monday, depending on how many notes you get done. Remember, there is that remind that I do want you guys to read that article and maybe check up on how it's going with that technology. And syllabus statements. Syllabus statements. Okay.